Hey team, I just wanted to send out this quick video for the last minute things you want to look over before the AP exam on Monday. Wanted to make sure that I was able to just give you my last bit of advice. So I looked over at the long FRQs, and the one thing that I noticed is that remember, in the long FRQ, A is always just something you should know that's not related to the question at all. So a lot of you answered what the PAL enzyme did specifically, and you did not answer the question about how enzymes increase the rate of a chemical reaction. So you answered what the PAL enzyme does, but not how enzymes are increasing a rate. So the correct answer was looking for lowered activation energy, because remember, enzymes are proteins that bind with a substrate at the active site. The substrate then is either broken apart or put together to make a product. And this happens during the enzyme substrate complex. So you had to have something either about the active site acting on PAL, or that PAL enzyme lowers activation energy, causing the reaction to occur. So something about the reaction occurring because of the enzyme was what it was looking for, and not just like what PAL does. So it was not looking for like PAL turns on and makes pigment production. So just remember that on the long FRQ, the both of them, A should be just knowledge that you have from this course, not necessarily related to the questions. So just remember that enzymes catalyze reactions by lowering which means to speed up by lowering the activation energy. And this was a slide that you would have seen had I been there for this unit, that you need to tattoo this on your face, that the activation energy for a chemical reaction to occur, remember, to break those bonds requires energy. And what an enzyme does is it moves the substrates so that they're in the correct orientation so that it can then have the reaction occur. So thanks, enzyme. The other thing that I feel like I had mentioned before was protein folding, which is a big part of Unit 1 that I think is the most strugglesome topic that I wasn't there for, so I wanted to cover that. A chemical binds to a protein composed of a single polypeptide chain and prevents the formation of an alpha helix that is typically formed in the absence of a chemical. Which of the following best describes the effect the chemical has on the structure of the protein? So if you were looking at an alpha helix, that would definitely be the secondary structure. So the secondary structure, which is held together by hydrogen bonds. So the primary structure, so first, just what are the parts of our proteins? So proteins are made up of amino acids, so individual amino acids joined together to make up a protein. And so there are three parts to an amino acid. There's the amino group, which is what gives it its name. So it has nitrogen. There's the side chain, which gives it its properties. And then there's the carboxyl group, which usually gives it a pretty good charge or makes it a little bit acidic, which is really nice. So we have, this is our general structure of amino acids. We put those together, we get a protein. So the primary structure of a protein is just the order of the amino acids. So it's just the nitrogen binding to the carboxyl group in a dehydration reaction. So that is how you get this setup of these chains. Another thing I wanted to mention on this slide is just to remember one of the questions they'll probably ask you about is the chemical composition. So knowing that proteins are CHON, carbon, hydrogen, oxygen, and nitrogen will be really important. And that proteins don't generally contain phosphorus. So the primary structure is the order of the amino acids, and then that's like just the carboxyl binding to the amino groups in that dehydration reaction, and it's just a single chain that's not folded. So then when you start folding it, you fold it first into its secondary structure. The secondary structure is either a beta pleated sheet or an alpha helices. So there's only two secondary structure options. So you either coil it like a pig's tail, or you fold it like you would fold a paper or like a fan. The beta pleated sheet and the alpha helices are held together by hydrogen bonds between the amine group and the carboxyl group. So the oxygen here is going to be negative because it's of the polarity, right? Carbon and oxygen form negative, or oxygen's partially negative because it is more electronegative. Nitrogen and hydrogen is partially positive because it is less electronegative. So this positive and negative come together to form that nice hydrogen bond. So these hydrogen bonds form, and when they do, they tend to make like either the alpha helices or the beta pleated sheet. And that's the only bond type in secondary structure. So that's the only one that can be broken in the secondary structure. The tertiary structure is between R groups, and there's more than one type of bond in that. So the R groups vary. There's several different R groups. So these are all your R groups that we looked at when we did that activity where you put the amino acids on the circle. So you have some that are polar. So you have some that are polar, you have some that are non-polar, you have some that are acidic and some that are basic. So all these different properties. Again, you'll notice that there's some nitrogen, there's some sulfur, but you're not seeing any like phosphorus or anything like that. So these R structures then stick out and they start to bind with each other. So like things that are hydrophobic are going to come be attracted to each other to avoid the water. And then things that are positive or negative that have like an ionic bond to them or an acidic or a basic charge are going to come together to form this ionic bond. And then these disulfide bridges are going to form between two sulfurs. So you have hydrogen bonds in tertiary folding, but you also have hydrophobic interactions, disulfide bonds, and ionic bonds. So all of those are involved. So then in this question, it asked about the additional hydrogen ions for an enzyme. And the additional hydrogen ions are, well, it asked about lower pH for an enzyme. And remember, low pH means more hydrogen. So there'll be an additional hydrogen bonds, and then those are going to disrupt the hydrogen bonds in the secondary, tertiary, and quaternary structures of the enzyme. So hydrogen bonds are not in the primary structure. The primary structure is peptide bonds. That's what holds those amino and carboxyl groups that happen via dehydration. The hydrogen bonds are in the secondary, tertiary, or quaternary structures. And quaternary is just when you bring two tertiary structures together. So this question would have been that answer. So I just really wanted to make sure that you covered protein folding. So you really want to make sure you review that because we're continuously getting those questions wrong, and that'll definitely be on the test. Also, chaperonins are assistant in protein folding, just in case you happen to see that word. You would see a large amount of them on like the endoplasmic reticulum. Well, good luck. I really hope that you guys are going to do an amazing job on Monday, and I can't wait to hear all about it on Tuesday. Good luck.